And away we go. All right, here we go, talking about, whoops, why is, oh, because I don't have the pen on. It's always something. Chromosomes, our favorite. All right, so of course, we're going to be more concerned with our eukaryotic chromosomes than our prokaryotic chromosomes, but make sure you know some of the differences. Okay, eukaryotic chromosomes are, of course, in a nucleus, while prokaryotic chromosomes are not. Eukaryotic cells always have multiple chromosomes. Prokaryotic cells only have one. Eukaryotic chromosomes are much longer and have much more DNA than prokaryotic chromosomes. And they are also linear, whereas prokaryotic chromosomes are circular. Okay, and all of these kind of differences together account for why eukaryotic cell division, i.e. mitosis and meiosis, is so complex, because it has to keep track of a lot more larger chromosomes. So I just wanted to go over this slide again, showing you how chromosomes are arranged. Here we have your DNA. Because there's a lot of DNA that needs to get folded up into the nucleus, we have some histone proteins that bind to the DNA and help to fold it up. You can see it folding here. And then once you have DNA plus histone right there, it is, of course, chromatin. And this is another difference between prokaryotic and eukaryotic chromosomes. Prokaryotic chromosomes do not have histones. They are not chromatin. Eukaryotic chromosomes do have histones. They are made of chromatin. Okay. And so for eukaryotic chromosomes, again, the foldiness just continues to fold until you get kind of that X structure that we typically recognize as a eukaryotic chromosome. And again, this chromosome that they have in the picture here, and here, and what I have drawn here is a duplicated chromosome, one that has already replicated its DNA. Okay, so here we have an unduplicated chromosome. Okay, so this chromosome has not replicated its DNA. Okay, this chromosome here has one chromatid. Okay, this chromosome here has two chromatids. And because it replicated itself and made an exact copy, the chromatids are identical. And we call them sister chromatids. Okay, and we got a little bit more to talk about the anatomy of a chromosome. So I suppose, kind of what we're starting to talk about here with these guys here and here is the anatomy of our chromosome. Chromosomes also have two other regions that are crucial to its structure and function. One structure is called a telomere, and the other structure is called a centromere. Okay, a telomere is the end of the chromosome. So since eukaryotic chromosomes are linear, they have two ends. And the ends are folded up into a very unique structure called a telomere. And this basically protects the end of the chromosome. Okay, so that's the telomeres are the ends, and the centromere is in the center, okay, of the chromosome, or near the center of the chromosome. And it has two principal functions. First of all, it holds the two daughter DNA molecules, which we also call sister chromatids. together after DNA replication and throughout most of mitosis. And also number 2, it is where the microtubules attach during cell division, mitosis and meiosis, to help move the chromosomes around and get them to the right spots for cell division. Okay, and the last thing we're going to look at here is just some more diagrams of chromosomes. Here again we have at the top, this is an unduplicated, so it has one chromatid. Okay, and here you can see the telomeres at either end, centromere in the center, and it has some gene loci here. Loci is the location of a gene on a chromosome. And then here we have a duplicated chromosome. So it has been replicated, its DNA, it's gone through S phase, or it's in a cell that has gone through S phase. And you can see now our duplicated chromosome has 2 chromatids, and they're called sister chromatids. And now we got, all these telomeres. Telomeres are always the end, and the centromeres in the middle. And you see here, the centromeres are holding the 2 sister chromatids together. So that's after DNA replication. And then once the chromosomes go through the cell division, the sister chromatids are separated, and they become independent chromosomes on their own. And that's it, I think. The end. Boom.